My name is Bob Lefkowitz. I'm an investigator of the Howard Hughes Medical Institute. I have been for many years, and a professor of medicine and biochemistry at Duke University Medical Center in Durham, North Carolina. Today, I'm going to be talking to you about my favorite scientific subject, receptors for hormones and drugs. I'm going to give two lectures. The first one will review some of the history of this field over the past 40 or so years that I've personally been involved, which has brought us to our current understanding of drug and hormone receptors. Then in the second lecture, I'm going to try to bring you up to date on some recent exciting research going on in my laboratory and in other laboratories around the world, which is changing in some very fundamental ways our understanding of how the receptors function and how they're regulated. Moreover, I'll tell you in that second lecture about how this new information can be harnessed to develop entirely new classes of pharmacological agents. The receptors that I'll be talking about today are known as the 7 transmembrane receptors, or also known as G-protein coupled receptors. In this first lecture, I'm going to review some of the history of how we have come to understand this remarkable family of plasma membrane receptors. Let me begin, though, with a few definitions. A receptor is a molecule on or in a cell with which a drug or hormone or a neurotransmitter initially interacts. A ligand is a compound which interacts specifically with the receptor. There are two main types of ligands: agonists, which stimulate the receptor, and antagonists, which bind to but do not stimulate the receptor. They're sometimes known as blockers. This little animation, which you see here, depicts an agonist interacting with a receptor and changing its conformation or shape so that it can signal. Two examples of agonists which work through this particular family of 7 transmembrane receptors are adrenaline and morphine. In this animation, an antagonist is shown in the form of this capsule, binding to the receptor but not changing its shape, and thereby not leading to biological signals. Several common examples of blockers or antagonists that you may be aware of are beta blockers, which is simply shorthand for beta-adrenergic receptor blockers or antagonists, antihistamines, or angiotensin receptor blockers, also known by the shorthand ARBs. Now today, I'm going to be talking about this remarkable family of 7 transmembrane receptors, or G-protein coupled receptors, as they're also known. The name 7 transmembrane receptor, of course, derives from their highly conserved structure, which we'll talk more about in just a few moments, which features a polypeptide chain which crisscrosses the plasma membrane 7 times. This remarkable family of receptors contains about 800 to 1,000 members and regulates virtually all known physiological processes in mammals and humans. In particular, many of the sensory receptors, such as those that mediate taste and smell and vision, are members of this family, as are so many drug receptors, such as those for beta blockers, antihistamines, glucagon, to mention just a few. But what makes this family of receptors even more important is that their central place in modern pharmacotherapy of human illnesses. It's said that somewhere north of 50% of all prescription drugs sold in the world today are drugs which function as agonists or antagonists for various members of this family of receptors. Let's take a moment to review some of the basic characteristics of how the 7 transmembrane or G-protein coupled receptors function. I'm sure many of you are familiar with this information, but I thought it would be a good idea to make sure that everybody is on the same page. So when one of these receptors, for example, the so-called beta-adrenergic receptor for adrenaline or noradrenaline, is stimulated by its agonist, it changes shape or conformation. This allows it to interact with a heterotrimeric G protein, in this case, GS. These proteins, of course, are referred to as heterotrimeric because they consist of 3 distinct subunits, in this case, termed alpha, beta, and gamma. The interaction of the agonist-activated receptor with the G protein is thought to dissociate it, or at least loosen the association of the alpha subunit from the beta-gamma subunit dimer. Then, both the alpha subunit and the beta-gamma subunits can independently function by activating various downstream signaling systems. Of course, the classical pathway is for activation of second messenger generating enzymes such as adenylate cyclase or phospholipase C, which would generate cyclic AMP or diacylglycerol. Now, although receptors has been an interest of pharmacologists for more than a century, quite remarkably, there remained great skepticism as to whether receptors existed in specific biophysical terms, as specific molecules in the cell that could be studied, until quite recently. In fact, I've put up here a quote from a very influential and very well-known pharmacologist of the 20th century, Raymond Ahlquist. Now, ironically, Raymond Ahlquist, a very decorated scientist and a winner of the Lasker Prize, amongst others, was the man who first suggested that there might be 2 distinct types of receptor for adrenaline and adrenaline-like molecules, which he referred to as alpha and beta-adrenergic receptors. This seminal contribution would ultimately lay the basis for the development of beta blockers by Dr. James Black. However, as late as 1973, Ahlquist himself wrote the following about the idea that receptors might actually represent specific molecules. He wrote, "This would be true if I was so presumptuous as to believe that alpha and beta receptors really did exist. There are those that think so, and even propose to describe their intimate structure. To me, they're an abstract concept conceived to explain observed responses of tissues produced by chemicals of various structure." Well, this quote from 1973 corresponds to approximately the time that I began my own independent work in my own laboratory at Duke. And it was clear to me and to others in the field at the time that if we wanted to bring these mythical receptors to life, to show that they really did exist as specific molecules, it would be necessary to develop a whole new set of technologies. And the first of these would have to be some form of radioligand binding. Now, by radioligand binding, we mean the idea that one could take a ligand, be it agonist or antagonist, radioactively label it, and physically demonstrate the binding of that radioligand to the receptor. If we could do this, we would have a way of studying the receptor's properties, studying how they were regulated, and perhaps a way of discovering new drugs, and most importantly, a means for following the receptors during purification efforts, which might allow us to isolate the receptors and thereby learn about their molecular properties. Let me give you an example of a radioligand binding experiment, the kinds of experiments that we did some years ago, and which led to some very interesting new information about how the receptors function. Shown in this top panel is a typical example of what's called a radioligand displacement curve. In this experiment, a radioactively labeled beta blocker was bound to beta-adrenergic receptors, which are coupled to adenylate cyclase, in the plasma membranes of cells. Then an unlabeled antagonist, such as alpranolol or propranolol, is allowed to compete at increasing concentrations and to block the binding of the radioligand. As you can see, this leads to a classical sigmoidal dose-response curve. We refer to this as being uniphasic. It has a hill slope of 1. It is Michaelian. In striking contrast, as shown in the bottom panel, if the same experiment is done with a beta-adrenergic agonist, such as isoproterenol, one gets a much shallower curve with a much lower hill slope, which can be resolved by computer fitting to 2 distinct affinity states, one of high affinity and one of low affinity. If one adds a guanine nucleotide, which will bind to the alpha subunit of the G protein, then as you can see, the curve is shifted to the right and steepened. And in fact, the affinity of this now steep or uniphasic curve corresponds to the lower of the 2 affinities shown here. A great deal of analysis of these types of curves taught us that agonists are able to stabilize a high affinity form of the receptor. This was set forth many years ago in the so-called ternary complex model. In this model, there's an initial step where the agonist, in this case depicted as H for hormone, binds to the receptor to form a low affinity initial hormone receptor or agonist receptor complex. When H is an agonist, a presumed conformational change in the receptor occurs, which at the time was hypothesized to lead to the binding of the HR complex to a 3rd component, hence the name ternary complex, leading to the formation of the high affinity HRX complex. When a guanine nucleotide, such as GTP, binds to this complex, it leads to its dissociation back to the low affinity state, and hence the marked shift in that curve. At the time the ternary complex was originally developed, it was not clear what X was. But the pronounced effect of guanine nucleotides on the stability of this complex immediately suggested that X might be the recently, at that time, discovered GTP binding or guanine nucleotide binding G protein. In fact, this model proved extremely useful over the years in dissecting such ligand binding curves and led to many insights. For example, the stronger the agonist in terms of its so-called efficacy and ability to promote signaling, the greater the stability of that complex and the higher the affinity of the high affinity complex. So this type of modeling proved very, very useful in developing models for how receptors were working together with G proteins. Now, once one had in hand such radioligand binding methods, it became possible to approach the difficult issue of trying to solubilize the receptors from the plasma membranes where they exist and beginning the difficult work of purifying them. It's important to understand that members of this family, the so-called G protein couple, the 7 membrane spanning receptors, are present at extraordinarily low concentrations in the plasma membranes of cells and would require several 100,000-fold purification in order to actually isolate them away from the other proteins. This is obviously an extremely difficult task and one which occupied my laboratory for many years. As our models, we used the adrenergic receptors, the receptors for adrenaline and related molecules, and focused on one in particular, the so-called beta-2 adrenergic receptors. The technique that proved up to the job, so to speak, and which was so difficult, was the technique of affinity chromatography. Now, in this technique, if one is trying to isolate a molecule, in this case the receptors, and one has a specific ligand, and we had several for that receptor, one takes the ligand, in this case that would be a beta-adrenergic antagonist, such as alpranolol, and covalently couples it to a solid support, such as a Sephiroth bead. One can then make a column of these beads containing the bound ligand, and if one has succeeded in solubilizing the receptors, which we were able to do after much trial and error with detergents, we were able to then apply the solubilized plasma membrane preparations containing the receptors but also many, many other proteins, to the columns of these beads, and now the receptors would stick by virtue of their specific binding affinity for the bound ligand. One could then wash the columns extensively to remove other proteins, and then finally elute the receptors with another ligand, such as alprenolol itself or any other beta-adrenergic antagonist or even agonist at high concentration. In doing this, one could achieve very, very high purification in a single step. One could then combine this with a variety of other more conventional chromatographic methods to hopefully isolate the receptors. We were able ultimately to succeed in purifying to homogeneity essentially all four of the then known subtypes of adrenergic receptors, called alpha 1, alpha 2, beta 1, and beta 2. And we did this by virtue of creating specific affinity chromatography columns in which we covalently linked either beta or alpha adrenergic antagonists to these Sephiroth beads. This effort took about a dozen years in my laboratory and devoted work of many students and postdocs. Ultimately, we succeeded in purifying all four of these subtypes to homogeneity. This is a slide I used to enjoy showing back in the 80s because it essentially reduced to a single frame the work of many, many students and fellows over more than 10 years. And what it is an SDS polyacrylamide gel on which we have run 3 of the 4 subtypes of highly purified adrenergic receptors. As you can see, each seems to migrate through the gel as a single polypeptide chain of about 60,000 Daltons. In each of these cases, the purified protein, the isolated molecule, appeared to bind radioligands and other adrenergic compounds with exactly the specificity and the stereospecificity that one would expect for the individual adrenergic receptor subtype. At this point, and this would have been the early 1980s, I would say, we thought that these kinds of conclusive findings would convince the skeptics that the receptors really did exist as specific isolated molecules. But in fact, the skepticism persisted in some quarters because it was said, well, how do you know that this isolated molecule, even though it can bind your drugs with the right specificity, how do you know that it's really the receptor, that it's really capable of conveying responsiveness to an isolated system? And to take that final step, we turn to the technique of receptor reconstitution. So in this method, we took our purified receptors and reconstituted them into phospholipid vesicles. We wanted to now take a system which was unresponsive to adrenaline and show that if we could insert this isolated molecule, the putative receptor, into that system, we could convey responsiveness to the adrenaline. So what we needed was a cell that didn't have the receptors. Well, that turned out to be a difficult problem. It turns out virtually all mammalian cells contain beta-2 adrenergic receptors. However, by trial and error and searching, we found that erythrocytes from an amphibian, the African clawed toad, Xenopus laevis, lacked beta-adrenergic receptors and hence did not respond to adrenaline, although it did contain the other components of the adenylate cyclase system, the G protein and the adenylate cyclase itself. It also contained prostaglandin receptors, and hence the adenylate cyclase of this cell could be stimulated to make cyclic AMP by prostaglandins but not adrenaline. So we took these cells lacking the beta receptor and we mixed them with these phospholipid vesicles containing the receptor. The vesicles fused with the plasma membrane of the cell, thereby inserting our putative receptors into the membrane. And lo and behold, the cell then became responsive to adrenaline and its rate of cyclic AMP production increased then in response to adrenaline. And so the last skeptics finally yielded and agreed that this molecule, which could both bind beta-adrenergic compounds with appropriate specificity and to convey to a previously unresponsive cell responsiveness to adrenaline was in fact the beta-adrenergic receptor. Once we had validated that the molecules we had isolated, and again, in these days, back in the 80s, we never had more than about 25 to 50 micrograms of purified receptor in hand at any one time. But once we had rigorously validated that these molecules were the receptors, we were ready to begin the difficult work of cloning the genes and cDNAs for these molecules to try to find out about their primary amino acid sequence. And after several years of difficult work, in 1986, we reported the cloning of the beta-2 adrenergic receptor. Now, this was a bit of a landmark in the field because to that point in time, no other G-protein coupled receptor had been cloned. The only exception was the molecule rhodopsin. Now, let's talk a little bit about rhodopsin. Rhodopsin, the so-called visual pigment, is, if you will, a receptor for photons of light. When photons of light are captured by retinal bound to the opsin protein, it isomerizes, changes the configuration or conformation of the rhodopsin, and signals are emitted which actually activate a G protein in the retina called transducin. Now, back in the mid-80s, most of us working in the signaling field were aware of the fact that there was a great deal of functional analogy between the visual system and the beta-adrenergic receptor stimulated adenylate cyclase. Both worked through a G protein, GS in one case, transducin in the other, and both worked through an enzyme, either a cyclic GMP phosphodiesterase in the retina or an adenylate cyclase in other mammalian tissues, which responded to the G protein. But nobody really thought that there was any structural analogy between rhodopsin and all the other members of the G protein coupled receptor family. However, when we succeeded in 1986 in cloning the beta-2 adrenergic receptor, we immediately could see its structural relationship to rhodopsin. Like rhodopsin, it had 7 stretches of hydrophobic amino acids, presumably representing helical transmembrane domains. As shown in blue, many of the residues in the transmembrane domains were identical, homologous with rhodopsin. There were, of course, connecting external and internal loops, many sites for regulatory phosphorylation on the C-terminus as in rhodopsin, and toward the amino terminus consensus sites for N-linked glycosylation. When I lectured today about the discovery, more than 20 years ago, of the close structural relationship between the beta-2 adrenergic receptor and rhodopsin, people are always amazed that we were all so surprised to find this. How could you not be thinking along those lines at the time? You knew about the functional analogies. This is true. Yet, as the contemporaneous literature reflects, none of us ever expected that there would be this close structural relationship. I think perhaps part of the reason for that had to do with even some prior history. The structure of rhodopsin, which is a remarkably abundant protein, it's about 90% or more of all the protein in isolated retinal rod outer segments. Rhodopsin it was available in large quantities. And when its structure was determined in 1982, it was originally, its sequence was determined not by cloning, but rather by classical Edman degradation, protein sequencing, because there was so much protein available. It was found to have a structure consistent with, suggestive of, 7 transmembrane domains. At the time, only one other such protein was known. That is, one other protein which had these 7 transmembrane domains, and that was bacteriorhodopsin. Now, bacteriorhodopsin is a protein present in very primitive archaebacteria, which is a light-driven proton pump. Now, when the structure of retinal rhodopsin was determined, and it too was found to have 7 membrane spans, it was very reasonably speculated by both groups who had succeeded in getting that sequence that the common feature of bacteria rhodopsin and rhodopsin were that they were light-sensitive proteins. And so it was put forth as an hypothesis that the 7 membrane spans must be a signature structural feature of light-sensitive proteins, which seemed very reasonable at the time. Only with the cloning of the beta-2 adrenergic receptor in 1986 did the idea first begin to emerge that no, perhaps this was the signature feature of the much larger family of G-protein coupled receptors. Over the next several years, we succeeded in cloning a total of 8 adrenergic receptors, as well as several other G-protein coupled receptors, and then others began cloning these receptors. And so it became very clear that in fact 7 membrane spans was the key structural feature of essentially all the G-protein coupled receptors. As I mentioned before, today there are almost 1,000 members of this huge gene family. They fall into several categories, sometimes referred to as A, B, and C. Most is known about the A family, which contains rhodopsin, the olfactory receptors, and the adrenergic receptors. Peptide hormone receptors generally fall into family B, and glutamate receptors and many others are in family C. Now, one interesting consequence of the very rapid cloning in the late 80s and 90s of dozens and dozens of these 7 transmembrane receptors was the development of the concept and the acknowledgement that there was a large number of so-called orphan receptors. So orphan receptors are sequences which seem to encode 7 transmembrane receptor-like proteins for which a functional ligand is not known. To date, there are currently about 600 or more of the roughly 1,000 members of this family are orphans. Now, of those, about a half, 300 or more, are olfactory receptors. But the rest are receptors which presumably perform other functions, and in aggregate, this group of orphan receptors represents a huge opportunity for the pharmaceutical industry for developing new types of drugs. Now, once we and others had realized that this highly conserved 7 transmembrane structure seemed to characterize all the members of this family, the next question became trying to understand how this highly conserved structural arrangement mediates the 2 core functions of the receptors, namely binding of ligands with great specificity and activation of specific G proteins, also with great specificity. Several techniques in the late 80s and into the 90s proved quite useful in this regard. Of course, there was classical mutagenesis techniques, but there was also the approach of creating chimeric receptors, one that we took right from the outset, publishing our first study in this regard in 1987 and 88. The idea here was as follows. We had just cloned the first 2 members of the G-protein coupled receptor family, the beta-2 adrenergic receptor, shown here in red, and the alpha-2 adrenergic receptor, shown in blue. Not surprisingly, their structures were very homologous, with about roughly 50% sequence identity in the membrane spans, which is one of the most highly conserved regions. Now, both of these adrenergic receptors, of course, bind adrenaline with high affinity and bind many other structurally related chemical molecules. However, alpha and beta receptors do very different things. In particular, they activate distinct G proteins. Alpha-2 receptors activate GI, thereby inhibiting adenylate cyclase, whereas beta-adrenergic receptors activate GS, thereby stimulating cyclic AMP. And there are specific agonists and antagonists which bind with selectively higher affinity to one than the other. And so we created, using recombinant DNA technology, a number, about a dozen different chimeras, and about half a dozen of those are shown here. Now, as depicted, sequences derived from the beta receptor are shown in red, and those derived from the alpha-2 receptors are shown in blue. Now, of the 6 or so chimeras shown here, 2 in particular were quite informative, and those are shown here. You can see that in each of these chimeras, there's a very small amount of beta receptor sequence. Most of the sequences are in blue. So such a chimera, such as chimeric receptor 8, shown here, is almost all alpha-2 receptor, with just this little insert from the beta-2. And, not surprisingly, chimeras such as these two bound ligands with an alpha-2 specificity. But remarkably, they activated adenylate cyclase rather than inhibiting it. That is to say, this very small, discrete segment of beta receptor, derived largely from the third cytoplasmic loop, was sufficient to dictate which G protein was coupled, the specificity of G protein coupling, and hence of signaling. Well, subsequently, a great deal more work from our laboratory and many other laboratories around the world led to our current understanding of how the structure of these receptor molecules mediates these two core functions. The membrane spans themselves, especially the outer regions, as well as sequences in the extracellular loops and amino terminus, are responsible for binding ligands and determining the specificity of ligand binding. Cytoplasmic domains, especially those in closest apposition to the plasma membrane, shown here in blue, are responsible for determining the coupling to G proteins. Now, it was in the course of doing this type of work that we were led to a serendipitous discovery of a type of receptor molecule which we called at the time constitutively active mutant receptors. This work grew naturally out of the work I've just described to you, aimed at trying to determine what regions of the receptor conveyed which functions. Having learned that cytoplasmic regions, especially as I showed you those very close to the plasma membrane in the third cytoplasmic loop, were responsible for coupling to G protein, we were carrying out a program of mutagenesis, trying to mutate various residues right here in the slide in the carboxy terminal region of the third cytoplasmic loop of the alpha-1 adrenergic receptor. Some of the residues that were being mutated are shown here. It was our reasonable expectation at the time that this would lead to a loss of function because this region appeared to be so important for coupling to G proteins. But to our amazement, several of the mutations seemed to lead to a very different result. That is, a result in which the receptor itself was active in leading to generation of second messengers, even in the absence of a ligand. And we call that constitutively active mutant receptor. Conceptually, the idea is shown here. In the classical pharmacological concept, receptors can exist in two states, inactive and active, R and R star, which are viewed as being in free equilibrium with each other. In the absence of an agonist or stimulant, the equilibrium is viewed as being far to the left, with just a little bit of, perhaps a few percent of receptor in the R star or active conformation. Agonists are viewed as ligands which have selectively higher affinity for R star, which stabilize this form of the receptor, thereby driving the equilibrium to the right leading to signaling. As we'll talk about in the second lecture, these concepts over the last few years have been significantly revised, as you will see, with interesting consequences. Now, presumably, there are constraints, physical intramolecular interactions between different parts of the receptor, which in the resting or R state keep it in an inactive form and keep it from activating G proteins. The activated or R star conformation of the receptor, which is stabilized by agonists, is presumably one in which these intramolecular constraining interactions have been abrogated, thereby leading to activation. We envisaged that these constraining influences were also abrogated by some of the mutations that we had made. Of course, at the time, some 20 years ago, we had no idea what the structural basis for these constraining interactions were. What we already were thinking, based on our mutations, that at least one key point would be the exact region we had made these mutations, namely in the carboxy terminal part of the third cytoplasmic loop. Subsequently, it turned out that by mutating either this region of other receptors or a growing list of other regions, one could create constitutively active mutant receptors for many different G protein coupled receptors. And in perhaps the most interesting consequence of this discovery, over the years, it was found that a growing list of human illnesses was caused by such mutations, either inherited or spontaneously occurring in various G protein coupled receptor genes. The very first one to be discovered was a sporadic hyperfunctioning thyroid nodules. We know today that as many as 90% of all benign functioning thyroid adenomas, sometimes referred to as hot nodules because they produce thyroxine, that more than 90% of those are due to mutations in the TSH receptor, the thyrotropin receptor, on the surface of those thyroid cells, which leads to a constitutively active mutant form of that receptor and to those nodules and hyperthyroidism. There are also examples of familial precocious puberty in males due to such mutations in the LH receptor and a growing list of others. Quite recently, over the last year or two, the very first crystal structures of the beta-2 adrenergic receptor are beginning to emerge. And a little cartoon of some of the work from Kobilka's group on this is shown here. Although it's not well depicted here, work of other laboratories before, together with crystal structures such as that of the beta-2 receptor and that of those of rhodopsin, which have been available now over the past decade or so, have served to begin to delineate a so-called ionic lock, an interaction between residues toward the C-terminus of that third cytoplasmic loop of the receptors and another highly conserved ionic region at the cytoplasmic surface of the third membrane helix, a highly conserved DRY or so-called dry motif. And so an ionic interaction between basic residues on the third toward the end of the third transmembrane domain and acidic residues in the third cytoplasmic loop seems to help keep the receptors in this tightly constrained inactive conformation and is believed to lead to activation when it is abrogated by conformational changes. But much remains to be learned about these particular intramolecular interactions. And the search for structures of activated G protein coupled receptors is a very dynamic and active area of research right now. Now, in addition to the work in the 80s and 90s on trying to understand the very nature of the receptors, and how they function. Another very active area, and one which is carried over to the present day, was the discovery of a universal mechanism which regulates receptor function. And this is referred to as a mechanism of desensitization. Now, desensitization is a problem which has intrigued me, a scientific problem, throughout my research career. And I think one of the reasons is that it is about as clear-cut and fundamental example of probably the most pervasive physiological principle in biology, and that is homeostasis. That is to say, when cells are perturbed by almost any influence, in this case a stimulant, an agonist, very quickly after the signaling occurs and the change in cell biology occurs as a result of that stimulus, very quickly thereafter the cell engages multiple mechanisms which are aimed at returning the cell to its basal state, even though the stimulation or perturbation continues. Now, in desensitization, this refers to the fact that when you stimulate a cell with almost any agonist, for example, very quickly the results of that stimulus wane. This is a classical example of that for a G protein coupled receptor. It happens to be the angiotensin receptor, stimulation of which by vasoconstrictor angiotensin leads to the generation of the second messenger, diacylglycerol. As you can see in these cells, which are HEK293 cells, which have been transfected with an angiotensin receptor, when angiotensin is applied to the cells, there's a very rapid spike in diacylglycerol. But even in the continuing presence of the angiotensin, the levels wane so that by 3 or 4 minutes, they're essentially back to baseline. So this is a classical example of desensitization. And for many years, my attention has been engaged by trying to understand how that works. Turns out, of course, that there are multiple mechanisms. But one universal mechanism, which seems to function for essentially all the receptors, is shown here. And it is one which we discovered back in the 80s. It was also discovered in terms of the function of rhodopsin by workers in the area of visual science. As you can see here, using the example of the beta-adrenergic receptor, when it is stimulated, it leads through the G protein arm to activation of second messenger signaling, as we discussed a while ago. However, very quickly thereafter, the activated form, the activated conformation of the receptor, is recognized by a kinase, a specific enzyme which is a member of the family of G protein coupled receptor kinases known as GRKS, and which are numbered. So in the case of the beta receptor, for example, the activated form might be recognized by one of those kinases called GRK2. This leads to phosphorylation of multiple sites on the C-terminus of the receptor, and perhaps in the case of various other receptors, on other cytoplasmic loops. The phosphorylation of the activated form of the receptor then facilitates the binding interaction of another type of protein, called a beta-arrestin, which is a member of a wider family of arrestin molecules, to the phosphorylated receptor. The binding of the beta-arrestin then sterically blocks interaction of the G protein, thus leading to a waning of stimulation and desensitization of the receptor. Other mechanisms also exist for desensitizing the receptors. They're not quite as general. For example, there are classical feedback regulatory loops whereby an enzyme activated here through the G protein, pKa, the cyclic AMP-dependent protein kinase, can feedback regulate the receptors by phosphorylating it and leading to desensitization by a mechanism that does not involve the beta-arrestins. This slide, from work done about 30 years ago in my lab, shows a key experiment which led to our discovery of the G protein coupled receptor kinases and to our understanding of this whole mechanism. As I told you, I had been interested from the earliest days of my career in this desensitization phenomenon. And so, whenever we developed any new technique for studying the receptors, I always wanted to bring it back to the study of desensitization. And so, Around 1979 or 80, shortly after we had developed the very first photoaffinity probes for studying the receptors, we did the following experiment. We took some cells, we desensitized the beta receptors by exposing them to a synthetic agonist isoproterenol, then washed that away, and then photoaffinity labeled the receptors with the new tritium-labeled photoaffinity probe that we had developed. In fact, what we were able to demonstrate when we then ran those solubilized, desensitized receptors side by side with naive receptors, which hadn't been desensitized on SDS gels, what we observed was that the mobility of the desensitized receptors was actually retarded as compared to that of the non-desensitized receptor. The middle lane here shows the pattern of a receptor which had been exposed to the agonist, but in which desensitization had been blocked by an antagonist. This subtle change in mobility, which was initially confusing to us, eventually, though, revealed the secret. Workers like Ed Krebs and others had been describing at that time that covalent modification of proteins, especially certain membrane proteins, could lead to their retardation in SDS polyacrylamide gel electrophoresis experiments by virtue of some change in SDS binding. So this immediately suggested to us that perhaps in association with their desensitization, the receptors were being desensitized. Very quickly thereafter, by labeling the cells with P32, we were able to prove that, and then we were able to go on over the next several years to isolate the kinase responsible for this specific desensitization. At the time we called it the beta-adrenergic receptor kinase. It's now known as GRK2. Subsequently, we and others were able to clone a number of other G-protein coupled receptor kinases. The very next one that we cloned after BARK, which is beta-adrenergic receptor kinase, and which we call now GRK2, the very next one that we cloned in my lab was rhodopsin kinase. Now, rhodopsin kinase was an enzyme which had been studied contemporaneously in several other laboratories, such as that of Paul Hargrave, and which seemed to be performing a function with rhodopsin, which was quite analogous to that the beta-adrenergic receptor kinase was carrying out with respect to the beta-adrenergic receptor. And we were able to clone it after purifying the enzyme and found immediately that its sequence was quite homologous with that of the beta-adrenergic receptor kinase, suggesting that these then were the first two founding members of a new subfamily of kinases, which today we call the G-protein coupled receptor kinases. All of the members of this family contain a highly conserved tripartite domain organization in which a central catalytic domain, which is highly conserved amongst the various members, is flanked by two regulatory domains, which vary depending on the nature of the kinase. There are 7 members of the family, which are further sorted into 3 subfamilies. GRKs1, rhodopsin kinase, and GRK7 are quite restricted in their expression to retinal rods and cones, respectively. GRKs4 is also quite restricted and found in very few tissues. GRK2, the original bark, 3, 5, and 6 are quite ubiquitous in their distribution, but the ratios of the enzymes vary from cell to cell and tissue to tissue. John Tesmer's lab has solved crystal structures of several members of the family, initially GRK2, in complex with a G-beta gamma subunit complex, which binds to a regulatory pH domain at the C-terminus. Now, the discovery of the beta-arrestins flowed very naturally from our discovery of the GRKs. In the mid-80s, a graduate student in the laboratory, Jeff Benovic, was purifying bark and made the initially disquieting observation that the more he purified the enzyme toward homogeneity, in other words, the greater its specific activity for phosphorylating the receptors, the more it lost its ability to actually desensitize the receptor functionally in an isolated system. While we scratched our heads about this, an important paper appeared in the literature by Thomas Kuhn. He showed that an abundant retinal protein, which at the time was referred to only as 48K protein, or S-antigen, somehow worked together with rhodopsin kinase to deactivate rhodopsin. And accordingly, the protein at the time was renamed arrestin because it arrested signaling. This seemed like exactly the type of protein we might be losing as we purified our kinase, and the loss of which might explain why we were losing desensitization ability of the enzyme, even as we gained phosphorylation ability. Of course, we knew starting out that 48K protein, or arrestin itself, couldn't be the molecule we wanted because it was expressed only in the retina. Nonetheless, it seemed like a starting point, and it seemed like something that might resemble what we were looking for. And so I contacted Kuhn and obtained from him some of his authentic arrestin protein. And sure enough, Benevik was then able to demonstrate that when added at high concentrations to our experimental system, it restored the ability of bark to desensitize the beta receptor. Now, as I mentioned, we knew that visual arrestin could not be the molecule we sought. And our initial biochemical approaches to purifying a related molecule did not yield any success. However, again, we were lucky, and within months, Shinohara at the NIH was able to clone the cDNA for S-antigen, arrestin, and then reasoning that the protein that we were looking for would be not only functionally analogous with visual arrestin, but structurally homologous with it, we obtained Shinohara's clone, and sure enough, by low stringency screening, Martin Losey, then a postdoc in the laboratory, was able to clone a structurally very similar molecule, which we called beta-arrestin 1. Within a year or two of that, another postdoc, Hoved Tramadol, was able to clone another molecule, which we called beta-arrestin 2. Now, with authentic recombinant materials in hand, we were able to prove that the molecules were what we were looking for and that there was, in fact, great specificity. We set up two parallel systems. In one, shown in the top panel, we had rhodopsin, which had been phosphorylated by rhodopsin kinase. In the other, shown in the bottom panel, we had purified beta-2 adrenergic receptor, which had been phosphorylated by bark. Now, into these reconstituted systems, we added either visual arrestin or beta-arrestins 1 and 2. In the reconstituted rhodopsin system, visual arrestin was quite potent in turning off the signal, but beta-arrestins 1 and 2 were quite weak. In contrast, in the beta receptor system, beta receptor phosphorylated by bark, or GRK2, both arrestin 1 and 2 were very potent in turning off signaling to the G protein, GS, whereas visual arrestin, as we had already shown, was quite weak. So clearly, we had a small family of arrestins here, which were quite specific in their abilities to interact with and desensitize either the visual receptor, rhodopsin, or the adenylate cyclase coupled receptor, the beta-2 adrenergic receptor. Today, we know that there are 4 members of the arrestin family. As I mentioned, visual arrestin and arrestin 4 or X-arrestin, found respectively in retinal rods and cones. Beta-arrestins 1 and 2, which somewhat confusingly are also known as arrestins 2 and 3, are quite ubiquitous in their distribution and are found in cells throughout the body. Now, this last slide shows a picture of some of my former postdocs and fellows who gathered together some 6 years ago to help me celebrate my 60th birthday. This is about half the people who've trained with me over the years. I show this picture because it is really due to their devoted work over a period now of some 35 to 40 years, which has allowed me to tell the story that you've just heard. In the second lecture, I'm going to tell you about how some new developments in our understanding of the arrestins and GRKs is completely changing our understanding of how the receptors are regulated. And I'll also tell you how it is leading to the development of entirely new classes of therapeutic agents.